Modern oncology relies heavily on single-axis biological therapies. But when we throw a single weapon at highly adaptive tumor states, those therapies predictably fail. To solve this, we are evaluating two distinct mathematical models. The first is the ANOVA-AX neoantigen pipeline, which calculates the precise parameters required for an extrinsic T-cell-mediated attack. The second is the apoptotic signal dynamics model. This maps the intrinsic pathway, the internal mitochondria-driven suicide sequence programmed into every cell. Analyzing either of these pipelines in isolation creates massive systemic blind spots. A perfect immune response is useless if the tumor's internal death threshold is mathematically unreachable. One dictates external targeting parameters, while the other governs strict internal threshold limits. They are physically different mechanisms, but they operate on exactly the same target. True eradication requires us to weaponize the intersection of these two models. We have to identify specific metabolic mutations, like IDH1 R132H, that are vulnerable from both the outside and the inside. Biological resistance is at its core a mathematical problem. We must audit both computational pipelines to design a viable dual-axis therapy. This formula calculates the ANOVA-AX composite score, evaluating vaccine candidates via a weighted linear combination of immunological variables. Notice the weights. T-cell recognition and MHCI binding carry the heaviest loads, 1.2 and 1.0. A peptide failing to bind renders downstream processes irrelevant. This table compares our candidate metrics. Look at our top result. Rank 1 is the IDH1 R132H 9-mer peptide. It succeeds because its 9-amino acid length provides an optimal structural anchor fit. More importantly, it achieves an exceptionally high DAI of 0.829. Now look at the bottom. Rank 9 is TP53R175H, the most frequent mutation in human cancer. Despite adequate expression, its DAI is a critical 0.048. DAI measures wild-type versus mutant binding affinity. A near zero score means they bind almost identically. Deploying this rank 9 peptide would be disastrous. The immune system simply cannot distinguish this tumor peptide from healthy, wild-type self-tissue. Contrast that with the high DAI of the IDH1 target. At 0.829, the mutant binds 10 times stronger than the wild type, guaranteeing maximum immunological foreignness. High-expression targets are useless and actively dangerous to the patient if their differential agrotopicity cannot breach the body's baseline immune tolerance. Reviewing the output logs reveals a logical anomaly. A 9-mer KRAS peptide ranks inexplicably lower than its 11-mer counterpart. The 9-mer should bind better. This inversion is a stochastic artifact. The pipeline currently uses random IC50 simulations, proving we need to integrate deterministic predictors like NetMHC-PAN before moving to physical trials. We also need to examine the pipeline's statistical validation, which uses ELI-SPOT trial data to measure immune response. There is a critical coding error in the ELI-SPOT grouping logic. Because the dictionary uses identical keys for both IDH1 variations, the first entry, containing the highest response data, is silently overwritten by the second. Even after accidentally dropping the strongest response group, the analysis of variance, or ANOVA, still returned a massively significant F statistic of 47.40. But when we run a chi-square test on the exact same dataset, the result fails to reach significance, sitting at a p-value of 0.0758. This happens because the chi-square test converts continuous ELI-SPOT counts into binary responder or non-responder categories. Binarizing continuous data destroys statistical power, especially at low sample sizes. Underlying biological efficacy can be completely masked by improper data conversions or falsely altered by undocumented code collisions. Next, we audit the ordinary differential equations that govern tumor and immune cell interactions over a simulated 100-day cycle. This chart graphs the ODE dynamics. On day 0, the vaccine boost floods the system, instantly generating 50,000 new effector T cells. The efficacy is rapid. By day 30, the killing rate of these new effectors overtakes the tumor's growth rate, driving the local tumor population to 0. But watch what happens in the post-tumor period, from day 30 to day 100. The mathematical system continues to climb, eventually stabilizing at an equilibrium of 5 million circulating effector cells. This state is a severe safety signal. A perpetually activated immune system circulating 5 million killer T cells with no tumor target will attack healthy tissue. Mathematical models that predict 100% tumor reduction are clinically unviable unless they incorporate precise decay parameters or immune checkpoint regulation terms to shut the response down. We have to shift our framework from extrinsic vaccines hitting the outside of the cell to the intrinsic suicide mechanisms operating inside the cell. Cellular death isn't a slow, linear decline. It is a strict binary decision controlled by steep, Cas-based-driven mathematical functions. This chart maps the apoptotic decision surface using a sigmoid function. A tiny 6-unit shift on the x-axis drives Cas-base activation from 5% all the way to 95%. Because of this extreme sensitivity, a mere 20% initial oxidative damage is enough to push the cell past the critical 0.5 activation threshold. Once a cell crosses that 0.5 inflection point, it is irreversibly committed to die. The cascade amplifies, and the cell executes its own DNA, regardless of any subsequent interventions. We can map two distinct pathological states on this curve. In neurodegeneration, like Alzheimer's, the neurons are sitting at 86.3% activation. This massive activation is driven by phase coupling. Dying neurons leak reactive oxygen species, synchronizing apoptotic signals with neighbors, destroying entire localized circuits simultaneously. Now compare that to a cancer cell. The cancer state sits precariously at exactly 51.9% activation. Cancer cells don't turn off the death signal entirely. They survive by hovering mere fractions of a percent above the mathematical point of no return. Hovering at 51.9% intrinsic activation, the cancer cell is highly primed. It requires only a minute signal push to trigger a total biological collapse. The therapeutic window required to push the cell over the edge is a fractional signal increase of just plus 0.085. We deploy the Innovax vaccine to attack extrinsically, while simultaneously applying a pro-oxidant to push the intrinsic threshold past 0.100. The IDH1 R132H mutation is the mathematically perfect target. Its mutant profile specifically reduces NADPH production, artificially elevating baseline oxidative stress before treatment begins. IDH1 R132H already holds the rank 1 spot for extrinsic targeting. Its unique metabolic defect makes it the easiest target to push intrinsically. Before moving to trials, we must execute the data corrections, advance the 9-mer peptide, fix the dictionary collision, deploy deterministic predictors, and model the ODE checkpoints. True eradication is achieved only when we synthesize systemic immune training with the inherent, inescapable mathematical threshold